Okay, so welcome back to this next video in which we are discussing circadian clocks. Okay, right, so I apologise for the abrupt ending to the previous video. What we're now going to discuss is circadian clocks that are present within mammals. Okay, and of course the mammal that we're most interested in is going to be the human, but we think that the circadian timing system in mammals is the same pretty much between different examples of mammals. Okay, right, so the main example of a circadian clock then that we have found within mammalian cells is another example of a transcriptional translational feedback loop. And this transcriptional translational feedback loop is going to be present in most of the cells of the mammal's body, basically. Okay, and what's going to be much more complicated about the mammalian circadian timing system compared to the Drosophila circadian timing system is not the actual transcriptional translational feedback loop. That's going to be pretty much the same, okay, but what's more complicated is how you keep all of the different cells in a mammal's body in time, basically, with the light-dark cycles. How do they all stay synchronized with the light-dark cycles? Because it turns out that in mammals, every single mammalian cell is not capable of keeping its own circadian clock in time with the light-dark cycle. Okay, so instead what we have is two special structures up in the brain called the suprachiasmatic nuclei, okay, which are receiving information about the light-dark cycles from the eyes. Okay, and we'll talk about this in much more detail later. And they therefore keep their circadian clocks in tune with the light-dark cycles, and they then get all of the other peripheral cells of the body with this same transcriptional translational feedback loop present within them to keep their circadian clocks in synchrony with the light-dark cycle. Okay, and that's what makes the mammalian circadian timing system more complicated. In addition, what we'll then talk about right at the end is that this beautiful system that I'm about to paint to you the picture of cannot be the full story, okay, because there are cells in the mammalian body which have circadian rhythms, but then cannot possibly have this transcriptional translational feedback loop that I'm about to describe to you in a lot of detail, okay, simply because they don't have a nucleus. So for instance, red blood cells don't have a nucleus. They cannot have transcriptional translational feedback loops, and yet they will have circadian rhythms, okay. So that must mean that there's another type of circadian clock, at least one other type of circadian clock that these cells which don't have nuclei can have in order to keep time, basically. Okay, and we'll then talk about post-translational oscillators, which are these types of circadian clocks which do not rely on a transcriptional translational feedback loop. And the most well-documented example of a post-translational oscillator is the cyanobacterial circadian clock. So we'll go to there to try and understand this in a bit more detail. So basically what I'm trying to say here is that the mammalian circadian timing system is complicated. Okay, I'm about to describe to you a nice simple transcriptional translational feedback loop that will look almost identical to the one that we've seen in Drosophila now, okay, and which is operational in a huge number of cells within the mammal, most cells within the mammal, okay. However, there must be other types of circadian clocks besides this one that I'm about to describe to you in at least some mammalian cells, if not in most of them. Okay, so our picture of this is incomplete as of yet. So what I'm about to do then is describe to you this transcriptional translational feedback loop that we have found, which is present in most mammalian cells in the mammalian body. Okay, and then we'll talk about how all of these different cells keep their transcriptional translational feedback loop circadian clock in tune with the light-dark cycles. Okay, right, so let's now talk then about this transcriptional translational feedback loop. Okay, right, so basically in mammalian cells there are proteins which are homologous to the proteins period in Drosophila and timeless in Drosophila. slightly more confusing. It turns out that in mammals we have multiple period proteins. Okay, so this is where things get a little bit more complicated. So basically in mammals we don't just have one period protein, instead we have 3 different types of period protein, and they all seem to do the same thing. Okay, and these are quite simply called period 1, period 2, and period 3. So all 3 of these proteins are going to do pretty much the same thing as the period protein that we saw from Drosophila did, and I think I'll keep the colour coding. So we'll have period proteins underlined once again in orange. Okay, right. Now, timeless proteins in mammals. Well, it turns out we don't have any timeless proteins in mammals, but we have proteins called different names which do the analogous thing to timeless proteins. Okay, and confusingly, these proteins are now called cryptochrome proteins. So cryptochrome proteins in mammals play the role of the timeless proteins in Drosophila. They don't do this thing about synchronizing each cell's transcriptional translational feedback loop circadian clock with the light-dark cycles. Okay, and in mammals you have 2 different types of cryptochrome protein, cryptochrome 1 and cryptochrome 2. Okay, so I want to stress again, these are analogous to the timeless proteins. Okay, they're going to form heterodimers with the period proteins, period 1, period 2, and period 3. Okay, right. So again, the transcriptional translational feedback loop circadian clock in mammals is going to involve the levels of these 5 proteins now, period 1, period 2, period 3, cryptochrome 1 and cryptochrome 2, going up and down on a 24-hour basis. Now, unfortunately, it's not going to be quite the same graph as I drew you for period and timeless in Drosophila. Okay, they're going to go up at slightly different times to where period and timeless went up in Drosophila. Okay, so let me now draw you another one of these graphs where I'm going to show you how the levels of period and cryptochrome vary over 24 hours. So once again, on the x-axis we'll have time, and we'll have it going from 0, 4, 8, 12, 16, 20, and then 24. Okay, so there's a full day. Okay, and once again, we'll assume that light is roughly from around 8 o'clock to 8 o'clock in the evening. I know that's not quite exact, but that's roughly where light time is and where night time is. Okay, right. And then what's going to happen in the case of the mammalian circadian clock system is that actually period and cryptochrome levels are going to be at their lowest at midnight. Okay, and they're going to be at the highest at midday, basically. Okay, so let me now draw this. So here we go, here they go right up to their top at midday, and then it's going to start coming down, and I'll try and get it roughly symmetrical. Okay, like so. Okay, so that graph there, which I'll colour in, I think, in orange, this is how the levels of period and cryptochrome proteins are going to vary during the day. They're all going to go up, all 5 of them are going to go up throughout the period from 0 AM to midday, okay, and then they're going to go down during the period from midday to midnight once again. Okay, so this is showing period levels, okay, and now cryptochrome levels, and I'll just put per/cry for period and cryptochrome proteins there. Okay, right. So now, again, what we want to see is what actually causes this. Okay, so we now need to see what are the analogous proteins to clock and cycle that we had present within Drosophila. Okay, so let's start off with the proteins that are equivalent to clock. So again, in mammals we have 2 clock proteins, essentially, 2 that are equivalent to one another and can both perform the equivalent role to what clock did in Drosophila. Okay, so once again we'll colour these important transcription factor heterodimers in green and blue. Okay, so remember in Drosophila, green was the clock protein, okay, and blue was the cycle protein here. Okay, now, in mammals we do have a clock gene, another gene called clock, okay, and this can, again, perform the same role as clock did in Drosophila, but we also have another protein that can do pretty much the same thing as clock could do, okay, well, can do pretty much the same thing as clock can do, okay, and this is called NPAS2. Okay, so this protein is effectively a very close homologue of the clock protein. Okay, so it's very close structurally to clock, and it can do pretty much the same thing as clock. Okay, so it can be part of this heterodimer in place of clock. So in this green slot here, you can either put a clock protein or you can have this NPAS2 protein. Okay, so that's that green protein there. So what then is the blue protein here? Well, that's the equivalent of cycle. Now, what is the equivalent of cycle in mammals? Well, it's a protein called BMAL1. Okay, right. So these are the important transcription factor heterodimers in the mammalian transcriptional translational feedback loop circadian clock. Okay, and we'll call this a BMAL1 clock, oops, I've missed out the MA, BMAL1 clock / NPAS2 heterodimer, okay, because it's consisting of BMAL1 and then you've either got clock or NPAS2 with it. So this is a BMAL1 clock / NPAS2 heterodimer. Okay, now, again, these heterodimers of BMAL1 with either clock or NPAS2, they are capable of binding to a special sequence of organic bases, okay, called the E-box within mammals. Okay, so I'll show this here. So here is the E-box that they're binding to, and these E-boxes will be present in the gene control regions upstream of certain genes, okay, and when the BMAL1 clock / NPAS2 heterodimers binds to the E-boxes, they will promote the expression of the downstream gene, okay, and once again, in these genes for the free period proteins and the two cryptochrome proteins, which are the equivalent to period and timeless in the Drosophila, you are going to have E-boxes, and therefore these BMAL1 clock NPAS2 heterodimers are going to drive increased expression of period and cryptochrome proteins. Okay, right, so now let's draw another one of those big pictures where we summarize the entire pathway then. Okay, so this time the point that it makes sense to start from is midnight, okay, which is quite a nice place to start from because that's the actual starting point of my graph here. So we'll this time start from 0 AM in the morning basically, and we'll see how this circadian oscillator is going to work. Okay, and again I'll stress that it is extremely similar to what happens in Drosophila. Okay, right, so let's start by drawing the nucleus once again here, okay, like so, okay, and then we'll have the period genes up here, and we'll have the cryptochrome genes represented down here, okay. So I'm not going to draw out 3 separate genes for all of the period proteins. In reality, the 3 different period proteins will of course have different genes. However, we will represent them all by this single one here. Okay, however, I want to stress that they do all have separate genes. They're not all one gene, okay? They are separate genes. Okay, so these are this picture here is representing the period 1, 2, 3 genes, all 3 of them. Okay, right. And down here, this picture likewise will represent both of the cryptochrome genes. Okay, so this is cryptochrome 1 and 2 genes. Okay, represented here in turquoise. And again, these genes, all 5 of them, will have E-boxes in their gene control regions. So I'll show these here. So here are the E-boxes in their gene control regions. And at 0 AM in the morning, we know that the levels of period and cryptochrome are going to be very, very low. Now, period and cryptochrome serve a similar function to period and timeless in Drosophila, okay, in that they inhibit the BMAL1 clock/NPAS2 heterodimers. So if we've got currently very low levels of period and cryptochrome because we're beginning at 0 AM in the morning, then that means that our BMAL1 clock/NPAS2 heterodimers are going to be free and they're going to be able to bind to our E boxes. So let's just put these in here. Okay, so here are BMAL1 clock/NPAS2 heterodimers. Okay, so here is BMAL1 represented in blue here. Okay, and then in green here, this will either be clock or it will be NPAS2, but it doesn't matter because NPAS2 is pretty much the same as clock anyway. Okay, right, so at 0 AM in the morning, what you're going to have is lots of free BMAL1 clock/NPAS2 heterodimers, okay, and they are going to be binding to the E boxes in the gene control regions of these 5 genes, the free period genes and the 2 cryptochrome genes, and they're going to be driving the transcription of these genes. So now, of course, what you're once more going to get is you'll get period mRNA being produced and cryptochrome mRNA being produced. So here, this is going to represent the mRNA for cryptochrome 1 and cryptochrome 2. Okay, so I'll colour that in once again in turquoise. Okay, and up here, this will be representing the mRNA for our 3 separate period genes. Okay, so period 1, 2, and 3 mRNA. Okay, so it's leaving the nucleus, coming into the cytoplasm of the cell, okay, and in the cytoplasm of the cell it will be translated into proteins. Okay, so now these mRNAs are going to actually become proteins. So here is our period proteins, and here are cryptochrome proteins. So once again, let's colour these in. So we'll have the period protein represented here in orange, as that orange box, and we'll have our cryptochrome protein represented as this turquoise box here. So this is either a period 1 protein, or it's a period 2 protein, or it's a period 3 protein, and this is either a cryptochrome 1 protein or it's a cryptochrome 2 protein. Okay, so let's just highlight that one up, cryptochrome 1 or 2, and this is period 1 or 2. Okay, right, so from 0 AM in the morning then, what you're going to be getting is the levels of period and cryptochrome in the cytoplasm of the cell are going to be building up and up and up. So if I bring back that important graph, okay, that's exactly what we're seeing is happening. Period and cryptochrome levels are going up and up towards midday. Okay, and then it's the exact same story that we saw in the case of Drosophila. Okay, there is then a lot of faffing about, basically. Okay, period and cryptochrome proteins, okay, any combination of them that you like. So you pick any one of the period proteins, pick any one of the 2 cryptochrome proteins, you can form heterodimers here between them. Okay, so here is our period protein here in orange, and here is our cryptochrome protein here in turquoise. Okay, so they've now formed a period cryptochrome heterodimer, so I'll just label this up. This is our period cryptochrome heterodimer. Okay, and this takes a long time to actually get these forming. Okay, so once again I'll colour these lines in blue. There's a lot of delaying about, a lot of faffing about. Okay, and then for the period cryptochrome heterodimers to actually go into the nucleus, again there is a lot of faffing about, basically. It takes a long time, and again the reason is that you need post-translational modifications to occur to the period proteins, to the cryptochrome proteins, in order for them to form heterodimers. Okay, and again you need post-translational modifications to occur to the period cryptochrome heterodimer in order for it to then translocate from the cytoplasm to the nucleus. So that's what is holding this process up, the need for post-translational modifications. Okay, now, as I did in the case of the Drosophila, I'm going to give you the names of a few enzymes that are important in making post-translational modifications, basically, to the period and cryptochrome proteins. Again, we don't know quite how it's orchestrated. Okay, we don't know exactly what order this all occurs in. All we know is some important enzymes that if you knock them out, it causes problems. Okay, so basically the enzymes that we know are extremely important in phosphorylating the period and cryptochrome proteins so that they can form heterodimers and so that they can go into the nucleus are casein kinase delta and casein kinase epsilon. Okay, so these are extremely important kinase enzymes which are capable of phosphorylating period and cryptochrome proteins, okay, and are important in the assembly of the period and cryptochrome proteins into period cryptochrome heterodimers, and then the translocation of period cryptochrome heterodimers into the nucleus. So they're part of this FAF in getting the period cryptochrome heterodimers into the nucleus, where they're actually going to affect their own expression, basically. Okay, right. Then, of course, what's going to happen is when they get into the nucleus, the period cryptochrome heterodimers are then going to inhibit the BMAL1 clock/MPAS2 heterodimers. Okay, right. So to go back to our graph, from 0 AM in the morning, you will have very low period and cryptochrome levels. We know that that means that the BMAL1 clock/MPAS2 heterodimers are free and active. Okay, they will therefore bind to E-boxes in the gene control regions of our period and cryptochrome genes, and the transcription levels of our period and cryptochrome genes are going to go up. So we're going to be getting more and more period and cryptochrome proteins actually being produced, so their levels are going up throughout the morning here. Okay, then there is this huge faff about getting them actually into the nucleus. Okay, and that takes ages, so you don't actually get them starting to go into the nucleus. You don't actually get period cryptochrome heterodimers getting into the nucleus until around midday, basically. Okay, then when they arrive in the nucleus, what they are going to do is stop their own transcription, basically, by binding to the BMAL1 clock/MPAS2 heterodimers. Okay, like so. So let me colour this in. So we'll have our period protein here in orange. Okay, we'll have our cryptochrome protein here in turquoise, and now it's bound to our BMAL1 clock/MPAS2 heterodimer here. Okay, and it's sequestering it away. Okay, and I should stress that not just one period cryptochrome heterodimer is going to be coming in. You're going to be getting lots of the period cryptochrome heterodimers coming in, and they will sequester away all of these BMAL1 clock/MPAS2 heterodimers. Okay, so that's what's happening at midday. Okay, all of the transcription factors are being sequestered away, they're being inhibited. Okay, and that means that you're going to put a brake on the transcription of all of the proteins which have E-boxes in their gene control regions, including the period and cryptochrome proteins, basically. So now production of more period and cryptochrome proteins is going to go down, and we know what will then happen. Okay, degradation is going to now dominate. Okay, now I want to stress degradation was occurring all through the up phase as well, but the production was greater than the amount that you were degrading, so you had net production, basically. But the levels of period and cryptochrome overall went up because production overrode the degradation, basically. Now we have turned production off, so now all we're going to see is degradation. Okay, so what's going to happen is the levels of period and cryptochrome proteins in the cytoplasm are gradually going to fall down and down as they're being degraded with no further production of them occurring. Okay, now, just like in Drosophila, even though period and cryptochrome levels are going down and down in the cytoplasm, there is a delay in that actually causing a reduction in the level of period and cryptochrome heterodimers within the nucleus. Okay, So, even though levels are going down and down in the cytoplasm, period and cryptochrome heterodimers remain in the nucleus, and they remain inhibiting the BMAL1 clock/MPAS2 heterodimers, okay, until right at the end. So right when you get down to midnight now, finally, when you're extremely low levels of period and cryptochrome in the cytoplasm, then finally something happens that then leads to the period and cryptochrome heterodimers in the nucleus being removed. Okay, and now we're back to the beginning. Now we have 3 BMAL1 clock MPAS2 heterodimers in the nucleus. Okay, we have very low period and cryptochrome levels, and what can now happen is the entire process can begin again. Okay, so you'll see that that is basically just exactly the same system as was in Drosophila. Okay, the proteins are a bit more numerous. We have some degeneracy, okay, and they're called different things, but in principle the system is exactly the same. Okay, again, it's an example of a transcriptional translational feedback loop. Okay, right, so we'll call it there for this video, and then in the next video what I'll talk about is, firstly, I'll talk about an accessory loop to this, because there is an accessory loop that's going to be stuck on the side of this system here, okay, involving a protein called Rev-erb-alpha, okay, and then what we'll do after discussing that accessory loop is we'll move on to the great question of how do we maintain all of the peripheral clocks in synchrony with the light-dark cycles when those cells don't have the capacity to do it on their own.